Hi, my name is Onatzevitch, and my project is an investigation of schizophrenia in human neurons. Schizophrenia is a neurological disorder that affects around 21 million people worldwide, causes hallucinations, paranoia, and delusions that can eventually become debilitating. Previous research into the disorder has identified changes in the brains of patients in animal models using postmortem tissue and animal representations of schizophrenia. However, the actual link between these mechanical changes observed in the brains and the symptoms of the disorder are unclear. The goal of my project was to investigate 4 changes that could possibly occur in the brain. However, instead of using models such as the previous research, animal models and postmortem tissue, to instead utilize stem cells taken from patients in order to generate neurons in culture that were never taken from their body, and then use those neurons to study changes in how the brain would function in those patients. So my project studied 4 different aspects of the function of neurons. The first was neuronal migration, which is a process by which neurons form in one portion of the brain and migrate to another as the brain develops. And a representation of this found that the schizophrenic cells moved on average 71% less distance than the healthy cells. The second aspect I looked at was the ratios of neural supporting cells to neurons. And we found that in the patient cultures, the ratio of astrocytes to neurons was significantly increased compared to the controls. The third aspect we looked at was the expression of genes related to the disorder. We found that genes that confer a very high risk for schizophrenia when they're mutated express themselves in different structures in patients compared to controls. And finally, we found that overall neural activity was significantly decreased in certain patient cells, but not others. So this project indicates not only a correlation between each of those 4 aspects and schizophrenia as a whole, it also reinforces that stem cell technology can be used to create representations of mental disorders without the need for animal models or postmortem tissue. Thank you.